So without further ado, I want to introduce our next speakers. We are so pleased to have Dr. Louise Markert here from Duke University and Dr. Audrey from the University of California at San Francisco. So please join me in welcoming them to speak about the thymus.
So I'm Louise Markert at Duke University, and what I do and have been doing for many years now is thymus transplantation in children that have a disease in which they're born without a thymus. And it's possible that thymus transplantation would be a therapy for APS. Now, the patients that I treat actually tend to have three main problems. They don't have a thymus, which means they don't have T cells, so they will die of infection usually within 2 years. Number two, they usually have a heart defect, and number three, they have a parathyroid problem, hypoparathyroidism. And I tell the families that I can't fix the hypoparathyroidism or the heart defect. The surgeons take care of the heart, but I can take care of the thymus. I can give them T cells that will work properly by giving them a thymus. And so, as I read all these words, I can't fix hypoparathyroidism and some problems that are existing, but we may be able to help future children avoid all those problems, and we may be able to help some children not get more problems. Now, I also should say, I'm a physician, I do this research, and who's ever heard of a physician who doesn't believe in what they're doing? So I want you to realize that, but I'll show you the data that I have, and I have hope that this will be able to apply to APS. I first will just go through my disclosures. So the thymus transplantation is investigational at this point. It's performed under an investigational new drug application with the Food and Drug Administration. I'm the sponsor of the investigations. And so I developed the technology since we started out in 1992. And Duke has now licensed the technology to a company called Enzivant Therapeutics, GmbH. And the purpose here is that we're going to get a biologic license so it will become a standard therapy. Portions of my salary and my research team's salaries are being paid by funding from the company, so we're helping them with the biologic license. And if the technology is commercially successful, both myself and Duke may benefit financially. So that's the disclosure slide. And now for APS-1. You all know much more about APS-1 than I do, although, I know a certain amount about it. But the symptoms relate to the autoimmunity that develops to many organs, including the ones that are listed here, and you've all heard about others earlier today and yesterday. So what is the role of the thymus in autoimmunity? The thymus is an organ that sits on top of the heart. It's like, I call it a schoolhouse. You go to Duke, everyone knows about the schoolhouse. That's where stem cells from the bone marrow go to the thymus to develop into T cells. And T cells, as you know, are white blood cells that fight infection, but they have to be properly educated. If they're not properly educated in the thymus, then the T cells can cause autoimmune disease. And part of the reason to not be properly educated is if there's not a defective AIR gene. So there are many cells in the thymus. I'll just mention the thymocytes are the developing T cells. The epithelium is sort of a structural type of cell that's in the thymus, but it has very important functions. And then there are dendritic cells, which are white blood cells that they're teachers, really, and they come from the bone marrow and go to the thymus to be a teacher. So this is so for the thymus to prevent autoimmunity, you need a good education. And I'll just discuss this a little bit. So here are the students. So those are the stem cells in the bone marrow. And they're going to go to the schoolhouse, which is called the thymus. And here, once the stem cells go in the schoolhouse, they're called thymocytes. Those are developing T cells. And when they leave the thymus, initially they're wearing a little graduation cap, and they're called naive T cells. And they're in the blood. We can pick them up on studies we do in the blood. Now, they're very important teachers that are in the thymus. Dendritic cells from the bone marrow that I just mentioned, they go to the schoolhouse and say, do not attack me. And they also work with the AIR gene to express various proteins from the salivary gland and the endocrine system, all these various systems where they're parathyroid, et cetera. So the dendritic cells are up here saying, don't attack me. And they're expressing some of these peptides and proteins. The epithelium itself, the structure of the thymus, also says, do not attack me. So any T cell that develops in the thymus is going to come out the other end if you have all the different parts of it, and it will be a wonderful T cell that will fight infection and not attack the recipient. So here's, again, the education. They're taught to not attack the baby, to be useful and fight infections, and they get a semester to study. It's about 4 to 6 months. And then they have to take a test on those two issues. If they don't get both answers correct, they have to die, okay? 90% of the T cells fail the test, so be glad you don't go to that school, okay? This is a tough school, but this is what the thymus does, and it does it very well. With respect to APS, the underlying problem is that there's a mutation in the AIR gene that's necessary for the thymus to teach the developing T cells, the thymocytes, to not attack the organs of the body. So it's a problem. The education is not working. So how might we prevent the autoimmune problems from occurring? Well, one possibility is to replace defective thymus with normal thymus. So here are some questions you might have. What is the experience with thymus transplantation in children in particular? And how is thymus transplantation done? As I mentioned, it's an investigational thymus transplantation, investigational treatment for children born with no thymus. At least that's what I'm using it for, children with complete DeGeorge anomaly. And because those children lack the thymus and hence don't have functioning T cells, all of them, as I mentioned, die by the age 2 years with rare exceptions from infection. So at Duke, we have transplanted thymus into 83 children with this particular disorder. 71% survive, and almost all of the deaths were from infection acquired prior to transplantation that killed the child before the good T cells could come out and start working. Because if they come with horrible adenovirus infection, it's really difficult to keep the child alive for the 4 months, or bad cytomegalovirus infection, it's really difficult. There's almost, if the child doesn't have an infection, the survival rate is just about 100%, and everyone who survives has a good immune system, maybe one exception. Okay. So where does the donor thymus come from that we use? Well, the donors are living children under 9 months of age who are undergoing heart surgery. And some thymus tissue is removed during some of the heart operations so that the surgeon can do the surgery, because it really sits right on top of the heart, right on top of the great vessels that often are defective and need repair. The discarded tissue is always put in a sterile container, which is handy. And we obtain informed consent from the donor's parents to use that thymus to try and help somebody else. And I might just add, really, there's only been maybe 2 or 3 refusals in the past 10 years to donate thymus when we've asked parents. We do routine testing on the donor and the donor's mother per FDA guidelines for transplantation. And the amazing thing is, for these transplants, tissue type matching is not required. Not at all. We can put any old thymus in, and we will get a beautiful immune system. And I don't know, are you all thinking what's the answer to this? Why this would work? A totally mismatched thymus could work? Okay. I don't see any hands waving, but okay. I do see hands waving. Why does it work? Oh, oh, I have one there, though. Right, well, there's no existing immune system, you're right, to attack the thymus, so it won't be rejected. But the reason it works is that as T cells are developing, they're taught by dendritic cells from that baby. So the baby's own dendritic cells go to the thymus and say, do not attack me. So the T cells that leave the thymus are not going to attack the baby, and they're going to fight infection in the context of that baby. So the dendritic cells in the blood, these white blood cells, are really critical to have it work. That's why we do not need to match. Okay, so here's a picture of what it looks like. When it comes from the operating room, it's just a little bit of tissue like this. It might be 4 grams, it's sort of small. We then bring it to the laboratory, and it is sliced into thin slices. And those are put on little filter papers that are about the size of a nickel in terms of the size. We have very thin slices of tissue. So this, again, is postnatal, up to 9 months of age, unmatched, and they're not related, at all. We culture them for 2 to 3 weeks. We change there's a nutrient broth in here, and we remove some of it every day and put more in every day, and it's in an incubator at body temperature. And then we do the transplant, and a general surgeon will take each piece of tissue, and they might have to cut it in half, and they'll transplant it into the thigh muscle. And so this is like planting tulips. Okay, so what happens is the surgeon will make an incision in the thigh through the skin, go down to the muscle, and then retract so you can see the surface of the muscle. That's like the surface of a garden. And then, just like planting tulips where you poke the holes in and then put the tulip bulb in and then put the dirt in, well, they use like a pair of forceps to just sort of poke the surface of the quadriceps muscle, and you make a few little pockets. It's not using a scalpel, just making little pockets. Put the piece of thymus in and then sew up the top so it won't squeeze out. And the little capillaries just grow in. And we use the quadriceps because it's a nice big muscle in a baby. It's not in the diaper area. It's in an area where you can have a scar. It's one of the places you're not going to see as much as other places. And I tell you, just like if you have a ball field, the players will come. If you have an empty schoolhouse, the students will come. And it's just, I'll never forget the first transplant. We didn't know if that was going to happen. And when we did the biopsy 3 months later, and the thymus was full of developing T cells, it was just an amazing thing. But they do come. And we have in the past, right now, we have a lot of patients, so and because it almost essentially always works, we were not doing it. But we used to biopsy at 2 months, and we could see normal-looking thymus in the quadriceps muscle, where it normally would not be. Okay. So here is the survival curve of the 83 infants, and so you see most of the deaths are from infections they came with, and so if we can get them through to 1 year, then they almost all just survive. And some of the deaths that have occurred in here are from like a cardiac arrhythmia after another follow-up heart operation, or a problem with a tracheostomy that one child had. There are a few things like that, but their immune system has been good. And so this is survival, so it's 71% survive, and the average age post-transplant of these 59 patients is over 10 years. And I have pictures on my wall of them, fishing and hiking and doing various normal family activities. Okay. So now a question for APS. Could thymus transplantation be done in children with APS who already have a thymus that's making T cells? And that goes back to your question. If you already have T cells, wouldn't you reject the thymus? That's what T cells do. Okay, so we believe that we could do this procedure, but of course it will need to be tested. I mean, I'm pretty sure, I think it's going to work, but we have to do more work, both with probably, I mean, I think we have quite a bit of work and we might be able to go to a patient. But at any rate, it would be best to do the transplant early before the autoimmune disease has affected a lot of organs, because it won't undo that. So it can prevent autoimmune disease but not fix damaged tissue. So first, how would this work? And then I'll show you some data that we have. This is not something totally straightforward. First, the child's own thymus would have to be removed, because that own thymus has defective teachers, and we don't want defective teaching. We want good teaching. So now the T cells, even though we remove the thymus, that doesn't remove all the T cells that would want to reject a new thymus. So we would give medicine, and we use, for the patients I have, an antibody that will kill most of the T cells in the child. And then we give immunosuppression with either cyclosporin or tacrolimus to suppress any remaining T cells so that we can get this donor thymus tissue into the thigh muscle. And we continue immunosuppression until new T cells develop and then stop it, which would likely be about 6 to 9 months. So what evidence do we have that this might work? Well, we have worked in rats. We've taken normal rats who have thymus. We have removed the rat's thymus, which we would do with a child, give an antibody to kill the T cells, and then put the rat on immunosuppression. And we leave the rat on, well, when we do that, all the T cells fall to 0, in particular the naive T cells. We keep them on the suppression for about 4 months, and by that time, they're making new T cells that are coming out of the thymus that say, do not attack me because of those good dendritic cells. And then we're able to stop the immunosuppression, and the rats have a good immune system. So here's my friend Louise. This is for the folks who do animal research. It's a Louise rat, but I call it Louise, okay, just for fun, okay. So here's this little rat who the thymus is removed and the T cells are depleted with antibody at time 0 from the experiment we did. And then we put in a thymus, a neonatal thymus from a different strain of rat, and continue immunosuppression with cyclosporin for 4 months, and then stop the cyclosporin. And then about 4 months later, assessing T cell development in the thymus. Here are some results. So this is naive CD4 T cells. So these are the T cells that are really educated. They're wearing their graduation cap coming out of the thymus. We can tell that they have. And we have one group of animals that has the thymus removed, the antibody treatment, cyclosporin, no transplant. And that's the sort of blue-gray line here. They start out with a certain number of naive T cells. After you remove the thymus and give the antibodies, there are no new T cells developing. I mean, they can't. They don't have a thymus. So if we compare that to a group that got the thymus, the red lines here are rats that we T cell depleted. They went down to 0. And then, just like in the human, it's very different than mice. Mice can make naive T cells in a flash. Well, rats are more like people, actually. And it takes more like around 3 months or so, and you see the naive cells increasing. And then we stop the cyclosporin, and they have their naive T cells. And so that is encouraging. And then with, let's see, so might it be possible to try this in APS? So this is really important. We are now working with Enzivant to submit that biologic license application for thymus transplantation, and this is critical to have this become a therapy that can be used across the country. So after approval of the biologic license, third party would likely pay for this treatment. I mean, just as now a bone marrow transplant can be used for DeGeorge, it's never been approved for use in DeGeorge, but all insurance companies pay for it. But at any rate, hopefully it won't be all that long for us to have this, but don't count your chickens. So a clinical research study would need to be done to show effectiveness for APS, but really if it's done in young children who are identified early, really only a small study would likely be needed to show whether it's effective or not. And I mean, of course, the FDA may want a variety of things, but this is sort of what I think to getting the license would be very helpful, and it shouldn't be that long. Now let's see. Oh, did I leave out a slide? Oh my God. I left out one slide. Okay, let me go back and look and make sure I didn't. Oh, this is a slide. I must have run right over this. I'm so sorry. So I showed you the rats, but how about in children? What data do I have? So here, oops, I guess that's the reason I skipped it. So we gave Thymus to a child who had T cells from a bone marrow transplant, but didn't have her own thymus, and she had very low naive T cells. So she was picked up, thought to have SCID, a severe combined immune deficiency, and was given a bone marrow and developed T cells. They were all paternal T cells. Or not, they weren't all. She had paternal T cells, and she had some of her own T cells, but just very, very low naive T cells. And various studies were done, and it showed that, in fact, it wasn't that she had a bone marrow problem, it's that she didn't have a thymus. She really wasn't a DeGeorge patient, but it was clear she didn't have a thymus. But she had a lot of T cells, and T cells reject grafts. That child also had severe autoimmune disease leading to absence of platelets, and platelets are part of the blood that make you clot. I mean, I spent like a full month in the clinic trying to get her platelets above a 1, which is like 0. So now I must say, this child had a beautiful, totally normal response to what we call a mitogen, showing T cell function. And myself and the whole group I work with thought, there's no way we're going to get this graft in to this child because of the presence of T cells that looked like they were pretty functional. But to our total surprise, the thymus transplant resulted in the child having naive T cells, a beautiful biopsy showing T cells developing in that thymus. And even though I told the family the autoimmune disease would not be affected, she no longer has any low cell counts. And she had very severe problems prior. So this child clearly is different from an APS-1 child. They're totally different. But both groups have T cells, which should reject a graft. And we were able to get the graft in and working in this child. And we were able to get it in the rat when we started with a normal rat. Okay, in Louise here. Okay. So at any rate, I feel this is something that can be considered or thought about, a potential therapy to help children who are born with problems with air, and help children who have, that we might be able to prevent future autoimmune disease. The big thing to realize is there does have to be that thymectomy early on, removing the thymus. It would be done with the thoracoscopic, where they put the tubes in and do the surgery inside. And we have some really good people at Duke who would be able to do that, and then follow it on with a thymus transplant. So something to be considering. So I want to just mention, you have all mentioned about the village. Well, I have a big village at Duke also. I have my laboratory group, and then it's like everybody else at Duke. So I'm in the division of pediatric allergy and immunology, the fellows and the attendings, the bone marrow transplant group, the attendings, all their staff, the nurses, pediatric general and cardiac surgery. Everybody is very much wanting to help in this, and other individuals too numerous to count. I could go on and on and on with the people who are participating, helping in this program. And then, of course, the funding we've had through the years, We've had NIH funding, we've had funding from the FDA, and right now we have funding from Enzivant, and we've had funding both from the departments of pediatrics and surgery. And without everybody's help, this never would have happened, but it has happened, and we may be able to extend this to APS-1 also. So thank you very much.
Okay. Hello, so my name is Audrey Parent. I'm a professor at UCSF. So I've been working very closely with Mark Anderson for the past 7 years on this project where we want to use human pluripotent stem cells to build a thymus, with one of the applications being for the study of APS-1. So as you probably all know by now, the mutation that affects APS-1 patient is in the AIR gene. And I won't go over the function of AIR because we've heard about it already, but so most of what we've learned is actually coming from mouse models. So they've been really useful. However, they don't fully recapitulate human disease. So there's probably some key differences between mouse and human that we still don't understand perfectly. So there's definitely a need for developing new models which would allow us to actually look at the human thymus. So the strategy that we decided to use to do that is to use human pluripotent stem cells. So to understand why those cells are so attractive for us, I want to point out two important characteristics. So first, why we really, they have so much potential is because they're what we call pluripotent. So that means that those cells can actually give rise to any cell type of the human body. So that's actually a pretty powerful thing. So I've put a few examples here of different cell types that we can actually make today from pluripotent stem cells, but there's many more, and in theory you could really use them to regenerate any of the cell types. Another important characteristic is that these cells can actually divide indefinitely. So you could think of making as many cells as you need to do your experiments. You can make millions of any of the cell type that you're interested in. So combine those two characteristics and you've got a really interesting system. So there's potential applications for human pluripotent stem cells. One obvious one being cell therapy. So there has been a lot of efforts in trying to develop protocols to make cells that would be transplanted back into patients. So for example, there's a lot of interest in regenerating pancreatic beta cells so they could be transplanted in diabetic patients. So that's definitely an interesting application. However, another one that's a little bit more recent is we now have the possibility to use those cells to model human disease. And part of why we can now do that is because there was a new technology that has been developed in recent years, and this is called induced pluripotent stem cells. So how it works is that instead of starting from a stem cell that has been generated from an embryo, so what we call embryonic stem cells, what we can now do is take cells from an adult. So we can take, for example, either from a healthy donor or from a patient that has a disease that we're interested in. For example, we could take an APS-1 patient. And what we can do is harvest some adult cells from that patient. So usually people do skin biopsy, or you can do a blood draw to get PBMC. So it's a non-invasive procedure. And then through a process that we call reprogramming, you can take that adult cell and bring it back to a state where it is pluripotent. So you can you bring it back in its differentiation state so that it now has the same characteristics where it can give rise to any cell type of the human body, and it can also divide indefinitely. So once you have that cell line, which we call iPS cell, what you can think of doing is then use your differentiation protocol that you've developed for an embryonic stem cell, and then you can actually make any cell type that you're interested in. Like in our case, we want to make thymic epithelial cells. But in this case, the thymic cell would have the genetic background of the patient it's coming from. So you could have a thymic epithelial cell that has the APS-1 mutation. And then once you have that cell, you can think of modeling the disease. So you can either do that in vitro, you can do it in a dish, or you can also do that in vivo. So you put back the cells into a mouse model, for example. And that could really help us understand the characteristics of the human disease. And then one last thing that you can think of doing, since you can actually make as many of those cells as you want, is to set up some drug screening to find molecules that can actually improve the function of your disease cells. Yeah, so that's one thing that's by itself really exciting. But if you now combine that with some other new technologies that have been developed recently, which are what we call genome editing technologies, that opens up a whole new avenue of research. So just quickly, how this works. So it's a technology that we call CRISPR-Cas9. So I won't go into too much detail, but it's a very powerful technique where you can now use an enzyme to cut the DNA, so you can cut in the human genome in whatever gene you're interested in. So for example, you could use CRISPR-Cas9 to target the AIR gene, and you can target it in a way that would create an inactive protein, so you would not be making any functional AIR. Or you can also use the same technique to introduce some specific mutation. So you can either start with a perfectly normal AIR gene and introduce mutations that you want to study. Or you can also think of doing the opposite, starting with a cell that has a mutated gene and use this technique to turn it back into a normal gene. So this is a technique that's pretty new, but we've been using it already for a while in the lab for different projects. And one of them has actually been funded through the APS-1 Foundation. And I won't show the data today, but I'm happy to report that we've successfully used the technique to inactivate the AIR gene. So we've decided to use this, the approach that's illustrated here on the left, where we made a mutation that will inactivate the gene. So we now have a stem cell line that does not express AIR. So we're looking forward to testing that cell line, making some thymic epithelial cells from it, and see how the loss of AIR expression actually affects its function. So now if we go back to our IPS slide, so what you can think of doing is combining the two technologies. So if you start with an IPS cell that has been made from a patient, you can actually now use genome editing technology to correct the mutation. So you could replace the AIR mutated gene with a normal one, and then you can potentially take this corrected IPS line and differentiate it using the same method, and then you would get some thymic epithelial cells that have functional AIR. And the really interesting thing about this is that this is coming from a patient, and then you could put it back, you could transplant it back without any immunosuppression, since it's perfectly matched to the patient. So it's a busy slide, but it's worth taking a minute to just think about how far we've come and what are the possibilities that these different technologies open up. So right now, so it's really a big picture summary of why we're interested in using the stem cells to make thymic epithelial cells and where we would like to be in a few years. But how do we actually get there? So the first part, where you make an IPS line, that's something that's well established. So people have made those IPS lines already. So I don't think that's going to be a problem. But where the roadblocks are right now, it's really in the differentiation method. So we've been working really hard on this problem. So personally, I've invested 7 years of my life trying to figure out how to make a thymic epithelial cell from stem cells. So we've made some progress. I want to show what we've done in the last years, but it's definitely a challenging issue to address. So how do you actually turn a stem cell into a thymus? So the way we think about this is, how does the embryo does it? So during embryonic development, what happens is that you start with a pluripotent cell that can make any cell type, and then you sequentially restrict the differentiation potential. So the cell will receive signal telling it, like, okay, you have 5 different ways you can go for the first step. You're gonna go specifically in that direction. So for example, in the case of the thymus, we need to make sure that it's not gonna be able to make a neuron anymore, not a cardiomyocyte, not a pancreatic beta cell. So you really need to force the cell into the right direction. And the way we do that is with chemical signals and also physical signals. So you need to try to recreate what's happening in the embryo, but in a petri dish. So in the case of the thymus, how is it actually made in the embryo? So I put an illustration here of the embryo with the gut tube in green. So the thymus is going to come from the gut tube. It's actually going to come from one of the most anterior part, so closest to the head. And then you see the final localization of the thymus is actually sitting right on top of the heart. So what we're trying to do when we do differentiations is to try to reproduce the signal that a cell that's developing in this anterior part of the embryo would see. So different factors to consider. For example, which signaling molecule do you want to test, when to add each factor, which concentration. So basically when we started this, there was no published protocol for making a thymus from human stem cells. So what we did is went through the literature and tried to find all the different molecules that had been shown to be important for thymic development, mostly coming from mouse studies. And then we selected a few and just tried a lot of different combinations. So that was a lot of cell culture happening. But what we came up with is this first version of the differentiation protocol. So we broke up the differentiation into different stages. I won't get into the details, but basically each stage is characterized by the specific combination of molecules. So usually activators or inhibitors of signaling pathways. And then to characterize each stage, we look at specific markers, which are usually transcription factors. So the main factor that we focus on is this FOXN1 transcription factor, which is the most specific factor for the thymus. And what we get when we do this differentiation, what you can see is that, so the final protocol is this step condition here. So we are getting some specific induction of FOXN1 using this protocol. We always compare with the tissue, the actual tissue that we want to make. So in this case, we compare fetal and adult, with the two tissues being actually pretty different because the thymus degenerate with age. But so it's something to keep in mind that the levels of expression of some of the gene will vary between fetal and adult. So in the case of FOXN1, we were getting some levels that are definitely lower than what you see in the fetal, but they were not that far from what you see from the adult. So conclusion from those experiments is it's not a perfect differentiation. So we're not getting a completely homogeneous thymic differentiation, but there are some cells in our culture that actually have the right markers. And we also looked at other markers like OXA3, which those had the right levels of expression when you compare it to both fetal and adult. So to get an idea of how heterogeneous our cultures are, we looked for different markers. So we like to do immunofluorescence. Ideally, we would have looked at FOXN1 directly, but I've tried really hard to find an antibody that works, and it was not successful. So instead, we looked at OXA3, which is also expressed in the thymus, and we look with EpCAM because we want to look at thymic epithelial cells. And then looking at a lot of different experiments, we came up with the estimation that most likely 15 to 20% of our cells are positive for those two markers. So keep in mind, it's not an homogeneous differentiation, but there's a significant portion of the culture that has the right phenotype. So to try to figure out if those cells can actually turn into functional thymic epithelial cells, we decided to move to an in vivo model. So the mouse that we chose to use is the nude mouse, which is it's actually a FOXN1 knockout. So these mice don't have a functional thymus. So because they don't have a thymus, they also don't have any T cells. But the interesting thing about this mouse is if you transplant functional tissue, you can actually rescue the T cell phenotype. So you can actually start making T cells that will go back into the periphery, and you can detect that in the mice. So we use the mice to transplant our stem cell-derived thymic epithelial progenitors, because what we're making in vitro is not a fully functional cell, so we call them progenitors. And then we also used in parallel some positive control. So keep in mind here that what you're looking at is mouse T cells being generated in a human thymus. It's not perfect, it works, but it's not perfect. And then in order to understand like this system, we found it was really important to have some positive control, some human positive control, which is the human fetal thymus. So what we did is put the cells under the kidney capsule. We waited for months. As Louise mentioned, it takes a lot of time to make some new T cells. So we grafted the cells, and then we actually bled the mice regularly to look for new T cells in the peripheral blood. And then once you actually start seeing those T cells being generated, then we harvested the graft and to look at what was going on. So to look for the epithelial cells, we use markers like the pancytokeratin antibody. That's the left panel here. it's kind of hard to see, but so in a normal thymus, in the control thymus, you see those big patches of red cells, which are epithelial. We could also see some of them in the TEP graph. So it's not as nice as the control. So it's mostly small islands of epithelial cells that we could detect. So it's not as good, but they're still definitely there after a few months into a mouse. And then we also look for more specific markers like cytokeratin 8, cytokeratin 5, which in a normal thymus, they segregate. So you have some regions that are more positive for K5, some that are more positive for K8, and some overlap. And we basically saw the same pattern in our little epithelial island in our TEP graph. We also look for T cells. So we stain for CD4, CD8. And what we could see is that in both the human fetal thymus graph and the TEP graph, you can find some patches of T cells. So they're not everywhere in the tissue, but there are some parts of the tissue where those cells are concentrated. So you can see them a little bit better here. So they are there. In order to get a better idea of the numbers, we did flow cytometry. So from some of the graph, we quantified by flow cytometry. And what you can see is that in both the HFT, so the human fetal thymus graph and the TEP graph, there are some double positive, which are the immature T cells. Not as many as in a wild-type thymus, so usually it's above 90% of the cells are double positive, so it's definitely lower than that, but there was some in our two human thymus graph groups, and there were none in our negative control graphs. So basically that was a key result for us, because it showed that even if we're not making a pure thymic epithelial culture, there are some cells in there that when you put them in vivo, they have the potential to turn into functional thymic tissue. So it's still not super efficient. So you can see some of the mice didn't work, but just the fact that there are some that works, that means that our differentiation protocol, the base that we start with is actually, it's actually good. So it's worth starting from there and optimizing so we get it to be more efficient. So as I said, what did we learn from those experiments? So the first thing was that was the confirmation that our differentiation protocol actually produced some TEPs that have the potential to turn into functional thymic tissue. So we should start from there and just try to make it better. However, there was some problems. For example, there was not a full maturation that we saw in vivo, including the lack of air expression, which was kind of disappointing. Multiple reasons that can explain that. One of them being that, as I said, we're looking at mouse T cells in a human thymus. So there's usually some crosstalk between the developing T cells and the thymic epithelial cells. So the fact that it's mouse with human might not be optimal. So what we decided to do is that we need to change a mouse model and move to what we call a humanized mouse model, where you're looking at human cells in a mouse. There was also, since we're working with heterogeneous cultures, what we notice is that in some of the mice, so this is an extreme example, but since we're not putting a pure population in, some of the mice had some outgrowth. So it's definitely not something you want to see when you transplant cells into a mouse. So conclusion from that is that we probably need to purify the cells before we put them into mice. So what we've been doing in the last years is try to optimize both the in vitro differentiation method, but also the way we do the transplantation. So the first thing we changed is we got what we call a reporter line. So it's a human embryonic stem cell line that has a GFP. So it's a green fluorescent protein that will be under its expression is under the control of the FOXN1 promoter. So what that means is that once we generate thymic epithelial cells in our culture, they actually will turn green, and we can detect that. So that has facilitated the optimization of the differentiation protocol, mostly because we can now quantify easily. So as you can see here, with this cell line, we can get up to 35% of thymic epithelial cells, which is better than what we had before. It's also good because we can isolate the cells. We can now do experiments with pure populations, which we could not do before. And then in order to apply what we learned with the FOXN1 GFP cell line to other cell lines, we also validated that you can use other surface markers to purify the cells. So for example, we're now using EpCAM. So what this shows here is that the green cells also express this EpCAM marker. So if you just enrich for those EpCAM positive cells, it's not going to be as pure as with the GFP, but you will enrich for the right cell population. So what we've been doing recently is we take our optimized differentiation cultures, and then we do an EPCAM or a GFP purification step. So we will enrich for those epithelial cells. And then once you purify them, we do a reaggregation step, so where you allow the cells to go back together, and then you form this little ball of tissue. So it's really, you can pick it up with a pair of forceps. It really is like a tissue. And that's what we put in the mice these days. Another modification that we've done is to add what we call a luciferase construct in the cell line, so that allows us to actually quantify bioluminescence. So once you transplant the cells, you can just use a compound that you inject in the mice, and the stem cell graft will actually produce light, and then you can detect that in live mice. So what we can do with that is that we can track stem cell survival without having to sack the mice every time. So it's been really helpful in trying to optimize the way we do the grafting procedure. And then just as a last piece of results, so those experiments are still ongoing, but what we've noticed with the mice that we've analyzed so far is that this enrichment step really helps with the overgrowth that we were seeing before. So it's kind of hard to see, but there's still some cystic structure, so there's a little cyst on the side there, but most of it looks much better than it used to when we were working with the heterogeneous culture. So definitely that additional step will help with the in vivo experiments. And then finally, the mouse model that we ultimately want to work with is this humanized mouse model. So we're currently using it's a new strain of mice, which we call NSG mouse. So these are immunodeficient. They don't have T cells, B cells, and K cells, so they don't reject the human cells that we put in there. But on top of it, they have a mutation in the FOXN1 gene, so they don't have an endogenous thymus, which means that if you see some T cell, once we do the co-transplantation with the stem cell-derived thymus, if you detect T cells in those mice, it has to be coming from the stem cell graft. While with the previous version of the humanized mice, if you could see some T cells, there was a slight possibility that it was coming from the endogenous thymus of the mouse. So it was complicated to analyze. So that took a while to set up this new mouse model, but now the mouse exists, and we've expanding the colony, and we're hoping to be able to do some experiments with those in the near future. So in conclusion, I would have liked to show a slide where we can make a fully functional thymus from stem cells. Unfortunately, we're not there yet. So we have made some significant progress in developing the differentiation method, but yeah, there is still work that needs to be done. The good news, though, is that with the genome engineering technologies, we now have an air-deficient stem cell line. So as soon as we have a good enough differentiation protocol, we will be able to make some air-deficient thymic epithelial cells from stem cells, and hopefully that will allow us to better understand the thymic defects that we see in the APS1 patients. And then finally, the beauty with those genome editing technologies is we can, even though it hasn't been done with the APS1 patient, people have done it for other diseases. We can now take a patient's cells, turn them into iPS lines, correct the disease-causing mutation, and turn those cells back into the cell type that was defective in the first place. So potentially, like once we optimize all of these and we combine all those technologies, there's a chance that in the future, maybe we can develop a cure for APS1 patients. Then the final slide, just to thank people who have worked on the projects. So mostly my two mentors, so Matthias Seabrock and Mark, which are both at the Diabetes Center at UCSF, some people from the lab. So it's been really a team effort developing the stem cell protocol and all the different assays to look at them, and also all the other lab members. And then finally the funding, and especially the APS1 Foundation for the funding to make the air knockout. Thank you.
Maybe a question that several people are thinking about, because ironically you started off by saying that your donor material was from children from whom the thymus was taken out, and yet the whole goal here is to restore the thymic function. So one of the big questions, I guess, is how long do we actually need a thymus for in order to develop appropriate immunocompetence? Maybe we don't have to restore the thymus lifelong. Maybe there's just a window that would be sufficient to give thymic function.
Well, first, I must correct the beginning part of your question. We use very small bits of thymus for our transplants. A full thymus may well be 25 grams, and we may be getting 4 grams. Yes. No, I understand the point. Okay, but I just had to say that. We're not taking all the thymus out of all these children, okay? In fact, the chair of surgery at Duke is an immunologist, okay? So he's very into keeping as much thymus in as possible. Now, you want the thymus to work as long as possible because you want it to be generating naive cells, new repertoire, and continuing to keep your repertoire very diverse as long as possible. Now, as we get older, though, our thymuses really do involute. And I don't know how long the thymuses in my patients are going to continue to make naive cells. I'm sure they'll involute sooner than a normal person who has a totally, a bigger thymus and all. But if you only give 1 dollop, the reason that bone marrow or cord blood transplants don't work as well in DeGeorge is you give them 1 shot of cells. You give them 1 repertoire on the day of the transplant, and every day thereafter they're losing some of that repertoire. So if you can instead give them thymus that will, at least for some years, are going to be making more new T cells, you'll maintain your repertoire longer. It won't be perfect, but it's better than just one dollop.
Yeah, but I guess what I'm really asking is there is clinical experience of children who had their thymus essentially removed completely by cardiac surgeons who may have slightly less immunological acumen than your colleagues. And what is the evidence for the amount of time?
Well, that is a difficult question. He's asking if someone takes all the thymus out of a patient, at what point do you run into trouble? Well, first, it's very difficult to take all the thymus out of a patient. It likes to spread, and some surgeons will try, and then they go back in for subsequent operation, and it's crawled all over the vessels like omentum. I mean, just a few cells that get out, they just divide and all. And when I called, because I was wondering, would this be a place where children would need thymus, I called major centers around the country. What problems do you have with immune deficiency from patients who had their thymus taken out? I tell you, they deny there's any. Now, there's one patient I may be transplanting who 18 years ago did have his thymus out. But in fact, I don't know if the patient was normal to begin with. And a problem with some of the published studies looking at thymectomy is they don't have any pre-data. And in fact, there's one paper that was looking at this, but if you go back to the previous reference where they actually did have a data drawn right before the thymectomy, before the heart surgery, well, they were just essentially all below the 10th percentile for age. So I think I don't think these thymectomies are full thymectomies, and there are a certain percentage of children that have heart defects that really do have a thymus problem as well. So I don't have the answer to your question. I don't think anybody does.
Hi, Dr. Markert. You actually transplanted one of our DeGeorge French-Canadian patients from Montreal many years ago, probably within your first cohort of patients. My question is about your use of that patient that developed an autoimmune disease after a bone marrow transplant as a proof of concept. I was just curious as to what autoimmune disease the patient had.
Well, I wasn't saying this is a perfect proof of concept. This was very severe thrombocytopenia.
Okay.
And in fact, and so that's different from
that's
APS.
very different. That's a B cell, and the only other autoimmune diseases I've seen, we have a huge bone marrow transplant clinic in my center, have been Graves and autoimmune thrombocytopenia, but that's antibody driven, right?
Yeah. Well, T cells often play a role in that if you don't have good T cells. But your point is well taken. But I was just putting it up there as a potential something I had not expected, that the autoimmune disease would resolve by me giving a thymus transplant. I said it wouldn't happen, and it did. So, things, I'm just showing you some of the data.
Yeah, I think one of the things that we forget is that there is a lot of talk between B cells and T cells. I'm not an immunologist at all. I'm an endocrinologist. But by virtue of the fact that acting with monoclonal antibodies on B cells can improve, to some extent, T cell diseases, I think is another proof that it's probably not just T cells, it's T cells and B cells, right? And maybe I'd like to hear more from the immunologists in the audience to talk about this aspect of APS1. I mean, look at your work on antibodies, Ole. I mean, how important are B cells in APS1?
Well, it's very hard to tell, but the only thing is that the autoantibodies seem to appear rather long before actually the disease develops, and in some way, I don't know if it's an antigen presenter or something even more elaborate that's going on. The other thing is the fact that they actually, when we have been looking, although it's not been systematic and extensive, they recognize the same autoantigen. And the reason for that, for me, is quite, I don't quite understand why you have autoimmunity regardless of MHC, always against 21-hydroxylase in adrenal insufficiency. And if you have APS1 where you have no MHC connection, it's 21-hydroxylase. If you have IPEX, you have GAD65 antibodies. So why isn't always the same? Out of 1,500 proteins perhaps, or even more, one single protein is selected. And for 21-hydroxylase, it's two non-amers that the T cells recognize, along probably at the same sites where the antibodies are binding.
One more question?
Well, let me be the first person to ask a dumb question, hopefully. Can you have two schoolhouses, meaning the bad schoolhouse that some of our kids or patients are born with, and then add a thymus in the thigh that produces good cells, and maybe, I'm sure the good cells and the bad cells fight and the bad cells prevail, but is there some milding or possibilities there?
Some experiments that we've done in the lab make us feel that we have to remove the recipient thymus, or we will never get the other one in.
Meaning it won't grow, or?
It'll disappear. You will not be able to find it. It's gone.
All right.
Okay. Sure.
Thank you, Louise and Audrey. Those were fascinating talks and really do help us open up our imagination to the possibilities of research for our APS1 patients. I can't thank you enough for making the trip to be here with us.